researcher meeting. My name is Rebecca Northeast, and I am a product manager here at Proteintech, and I run these ECR events. I'm just going to start with a quick company introduction before talking about how the event will run today. So if you're not aware of who Proteintech are as a company, we are a leading life science reagent company, and we sell products such as antibodies and immunoassays. So we have antibodies against 13,000 human targets, as well as various ELISA kits and IHC kits. And we also have recombinant cytokines and growth factors, which we sell through our Humankind brand, and also nanobody-based reagents, which we sell through our Chromatech brand. So today we have a really exciting lineup of speakers. So first off, we're going to have Professor Erik Musiek from Wash University at St. Louis in the United States. He's going to be talking about circadian clocks, glial function, and neurodegenerative disease. Next, we'll then have our chair, and she's going to be introducing all our ECR speakers. Our chair is Roisin McManus, and I'll introduce her in a minute. But then we will have the speakers, Jake Dundee, Danielle Beckman, Paige Mumford, and Ismail Edinieye. So our chair today is the fantastic Roisin McManus. She is a senior postdoctoral scientist and an Alzheimer's Initiative Project Leader at DZNE, which is a neuroscience institute in Bonn in Germany. She first got her PhD in Alzheimer's research at Trinity College Dublin in Ireland, followed by a postdoc when she moved to Bonn in Germany in Dr. Henneke's lab, and this was on the NLRP3 inflammasome. And now she works in Prof. Latzer's lab, and she is the leader of the initiative that I mentioned before, and she's currently working on immune signaling and how it influences microglial metabolic reprogramming and the meta-inflammation in neurodegenerative disease. So with that, I will hand over to Roisin, and she will introduce our keynote speaker, and I'm really excited for the event today. Thanks, Roisin.
Thank you, Rebecca. That was a really lovely introduction. First, I wanted to say welcome to everybody who's joined, and I wanted to thank Rebecca for putting this really wonderful symposium together. As anyone who's organized such events knows, it's a lot of work. So thank you, Rebecca, for bringing us all here today. Yes, so our first speaker is Professor Erik Musiek. Erik is the Charlotte and Paul Hageman Professor of Neurology at Washington University School of Medicine in St. Louis, and also co-director of the Center on Biological Rhythms and Sleep, also known as COBRAS. His lab focuses on how the circadian clock regulates neuronal and glial function and examines mechanisms linking clock dysfunction to Alzheimer's disease and other neurodegenerative diseases. Dr. Musiek is also a practicing neurologist who sees patients in the WashU Memory Disorders Clinic and at the Barnes Jewish Hospital. He completed his MD-PhD training at Vanderbilt University in 2007, neurology residency at the University of Pennsylvania in 2011, and a postdoctoral fellowship in dementia with research in the lab of Dr. David Holtzman at the Washington University in 2013. Erik will be speaking to us today about circadian clocks, glial function, and neurodegenerative disease. There'll be an opportunity for questions at the end, so please, if anyone has any queries, just pop them in the chat and we'll read them out and get Erik's immediate feedback on them. Thank you.
Thanks very much, Roisin, and thanks to Rebecca and Proteintech for a chance to speak at this great event. I'm excited to hear from a bunch of exciting up-and-coming leaders in the field and to take 25-30 minutes right now to try to convince all of you that circadian clocks and circadian rhythms might be important in some of the work that you do. So it may not be the first thing that pops to mind when you think of neuroinflammation, but hopefully 20-30 minutes from now, you'll think about it a little bit more. So I'll get started. So you probably all know that we have a circadian clock. It may not be something you think about a lot, but it plays a major role in all of our lives, how our bodies have adapted to the rotation of the Earth, and that we have a 24-hour timekeeper in all of our cells that helps to align, the cellular function and behavioral functions of an organism with the external environment. And I think the other thing the circadian clock does is it helps the body to anticipate changes in metabolic demand over the course of the day. So, there are certain sort of reproducible stresses on the body that happen as a result of the time of day. You eat breakfast in the morning, you go to sleep at night, you eat dinner at certain times, you're active during the day. And the circadian clock allows cells and organs, even behavioral adaptation to prepare the body kind of for those sort of stresses. And the immune system is certainly part of this process. So the immune system shows very pronounced circadian rhythms. And this is a diagram from Annie Curtis in Dublin that kind of illustrates some of the work in this area. I mean, I could speak about this for the entire hour, but there's been a lot of work looking at different inflammatory responses. This is mainly innate immune responses and how they vary over the course of a 24-hour day in mice. Now, it's important to remember mice, they have essentially the same circadian clock as humans, but they are nocturnal, so they sleep when we awake, so their phases are flipped. But otherwise, they have all the same clock genes and very similar regulation of a lot of these same pathways. So it turns out that in the evening for a mouse, when it's starting to enter into its active phase, the immune system is very active. If you give a mouse LPS or some other inflammagen in the evening, it can cause a much larger inflammatory response than if you give it to them in the morning. And that's the case for a variety of different pathogens and for a variety of different inflammatory responses. And it may be something that we don't all think about necessarily when we're doing our experiments, but obviously the circadian system is somehow governing inflammatory responses in general, although very little is known about the brain. And the way it does this is through this machinery. You're probably, many of you, familiar with. There's a molecular clock in almost all of our cells, essentially all of our cells in our body, that keeps time, even in the absence of any cues. And so there's a key transcription factor called BMAL1 that I'm going to talk about a lot today. It heterodimerizes with clock or NPAS2 to form a heterodimer that can bind to EBOX motifs throughout the genome, drive transcription of many, many genes. BMAL1 is sort of regulated in a rhythmic manner by its own negative feedback regulators. So it drives transcription of period, cryptochrome, and reverb genes that then feed back and inhibit this. And this whole system, this feedback loop, is tuned to a 24-hour period genetically. So if you take cells out of an animal and put them in a dish, they'll continue to have this 24-hour rhythm if you can synchronize them somehow. We can do that in a variety of ways. So this is a genetically encoded 24-hour clock that drives transcription that's in essentially all of our cells. And it really lies at the root not only of cellular rhythms, but of behavioral rhythms as well. So just a little more evidence of what I'm talking about. So this is some data, again, from Annie Curtis and several other investigators, showing that when you give lipopolysaccharide, a large dose, 25 milligrams per kilogram, so this is a lethal dose of lipopolysaccharide. If you give it to the mice in the morning, ZT0, that's lights on, so usually 6 a.m., you get about 60% or about 40% mortality. But if you give it to them at 6 p.m., lights out, there's almost 100% mortality after 6 days. If you disrupt the circadian clock by knocking out BMAL1, so that transcription factor, if you delete that, the entire clock breaks. The mouse becomes arrhythmic. Then all the mice sort of look like the 6 p.m. group. They all die in response to LPS. So it has a really profound effect on the response to LPS. It's not just LPS. We see the same thing with listeria infection, that the time of day, in this case, it's at lights on or in the middle of the day, the animals survive more or less depending on when they got this listeria infection, and there's differences in the amount of cytokines produced. This is all kind of peripheral work. But even in the CNS, Christoph Scheierman is really a leader in this area, shows that some of you are probably familiar with the EAE model of MS. So you immunize mice with myelin, with MOG or other CNS proteins, and then they develop an autoimmune response days later. But the Scheierman lab showed that the time of day at which you immunize the mouse, either in the middle of the day or in the middle of the night, that almost 2 weeks later, it has a big impact on the severity of the disease that they develop. So this is maybe surprising, but they go on to show that, you know, the mice that are immunized in the middle of the night actually have less severe disease. They survive better and they have less demyelination. And they've shown through many papers that this has to do with trafficking, right? Sort of within the first few hours after the immunization of immune cells and lymph nodes. So the time of day matters. We're interested in why, how do these clocks actually regulate some of these things? Because you can imagine if you have this clock, you can manipulate it somehow as kind of a way of altering the immune response in the brain or the inflammatory response in the brain. So many cells, as I mentioned, have a circadian clock. I'm going to talk a bit about astrocytes because we have sort of the most mature data on astrocytes, but astrocytes have really strong circadian rhythms. So if you transfect them with a per1 or per2 luciferase construct, and then you put luciferin into the cell media, if you give them a little serum shock or you change the media, they will show these really beautiful 24-hour rhythms in culture. So there's no light, there's no other Zeitgebers going on here. And actually, it peters out after a few days, but if you just give them another shock, they'll just continue to have it again. So essentially, all these cells are having circadian oscillations. It's just they become desynchronized over the course of a few days. And also astrocytes, because they're often touching each other, they can form sort of a syncytium. And these connexin proteins help to kind of keep for a more consistent circadian oscillation. So, we were interested in what is the circadian transcriptome in astrocytes. So what genes are being regulated by the clock in astrocytes? And does disrupting this clock have any impact on disease processes? And what does disease process do to this circadian oscillation? No one had really looked at this in vivo before that I know of in mice. So there is a group at Tufts that has done the same kind of work in flies a few years back. But fly astrocytes are quite different than mouse astrocytes. So we wanted to do it in mice. So we used a method called translating ribosome affinity purification, or TRAP. It's similar to ribotag. So we also used ribotag. Essentially, you just drive a tag of some kind, GFP or HA, with a cell type specific promoter. In this case, it's an astrocyte specific promoter, so that all the astrocytes in this mouse have a GFP tag on their ribosome. So it's RPL10A is a ribosomal subunit protein, has a nice GFP tag on it. You can sack the mice, you perfuse them with cyclohexamide to freeze all the RNA onto the ribosomes, and then you immunoprecipitate out your ribosomes that are tagged. So these are astrocyte ribosomes, and then you can sequence the RNA, and you can essentially look at all the genes that are being translated in astrocytes at a given moment in time. And this is all work done by Pat Sheehan. So I want to try to, because it's an early career investigator forum, mention some of the early career investigators from my lab. So Pat, really fantastic PhD student, is now at Dory Schaefer's lab in UMass, just starting his postdoc. So I'm sure you will keep an eye out for Pat. He's doing great things. So he did all this work. He crossed these trap mice either into wild-type mice or to APPPS1 mice, so a mouse model of amyloid plaques or a beta amyloidosis, Alzheimer's model. This is an aggressive model from Matthias Euchre's lab, and these mice get plaques, a lot of plaques by 5 months old, which is when we sack them. So we had wild-type mice at 5 months, APP mice at 5 months, and then Pat also took down a cohort of 22-month-old wild-type mice to look at the effects of aging on the astrocyte circadian transcriptome. And what Pat did each time is he put the mice in constant darkness for 24 hours so they don't have any outside cues, and then he sacked mice, several from each cohort, every 2 hours for a 24-hour period. Actually did this multiple times. Not the most fun experiment, but he's a trooper. And the data looks amazing. So we sequence these mice, and you can see the clock genes have, this is BMAL, this is one of the clock genes, this is cryptochrome 1, another clock gene. They have really nice oscillations. This is not normalized in any way. This is just raw CPM. Each dot is the average of 2 mice of a given genotype. So BMAL is supposed to be high at 6 in the morning and supposed to be low at 6 at night. So everything looks great. And interestingly, the circadian clock the core clock was not really impaired at all in astrocytes by the presence of plaques. So the mice that have plaques still have really nice circadian rhythms in their core clock, which is a little surprising to us because there's a lot of literature showing that circadian rhythms are disrupted in Alzheimer's disease. And the thinking was that the amyloid plaques might do it, but perhaps not in astrocytes. However, if you look more broadly at the whole transcriptome, you can see there's several hundred genes that are rhythmic. So we have an algorithm that can detect circadian rhythms in different transcripts. So each one of these is a time of day, each one of these is a gene. These are some of the rhythmic genes. You can see they're organized by when they peak. So these genes peak in the morning, middle of the day, at night. And many of these genes, several hundred of them that were rhythmic in the wild-type mice, this is in cerebral cortex tissue, lose their rhythms in the APP mice. So the rhythms are dampened such that they're no longer detected by our algorithm. However, it's not quite that simple. So there are actually a lot of genes who are not necessarily rhythmic in the wild type mice, but in the setting of amyloid plaques, they gain rhythmicity. And this is a phenomenon called circadian reprogramming, that somehow the clock seems to be able to change its rhythmic targets in the setting of certain kinds of stress. And we don't know how that works yet. It could be an epigenetic phenomenon, but we're not really sure. But it seems to happen in the setting of amyloid plaque. So even though the core clock is still oscillating with the normal rhythm, the target genes change. So let's talk about what some of those target genes are, because that's much more interesting, I think. So some of the hits that we get when we look at these genes that lose rhythmicity are actually amyloid beta is in there, response to radiation, which is sort of a cell stress, oxidative stress kind of pathway. A variety of different things going on that then become arrhythmic in the setting of plaques. So a couple examples, this is insulin degrading enzyme. So this is an enzyme astrocytes produce that can degrade amyloid beta. So if you have less of it, you actually accumulate more amyloid plaques. It's normally nice and rhythmic in wild type mice, and the red is the APP mice that loses that rhythm and is actually lower most times of day in the setting of plaques. CDKN1A is a cell cycle regulator that's upregulated in the setting of cellular senescence. It's actually one of the main markers of cellular senescence. So it's highly rhythmic. This has been shown by other groups, driven by the clock. But in the setting of plaques, it actually loses its rhythm and is sort of pegged in a high position. So, the time of day at which you're taking down your animals may have a big impact on whether or not you see transcriptional differences in these different genes. There's also genes that become rhythmic. So many of these are inflammatory genes. I just put CCL3 in here because it's a chemokine that's normally really not expressed in wild type mice, but in the setting of plaques, it becomes rhythmic. It actually increases massively, and it's still low, but it becomes rhythmic. And there's a number of chemokines that show the same sort of pattern. When they are upregulated in the setting of plaques, they have a clear circadian rhythm in their expression that you may not know if you're not thinking about it. Also, like there genes like clusterin, which is a major GWAS hit for Alzheimer's disease, has a really no rhythm at all under normal circumstances, but in the APP mice takes on this very nice rhythm, even though its levels don't necessarily increase. So in astrocytes, in the setting of amyloid plaques, the number of genes are actually the same that are rhythmic in the APP mice as opposed to wild type, but they're different genes. So there's about 81 differences, but the total number, 391 versus 308, doesn't really change much. So it's not like the plaques are disrupting the clock, they're just changing the clock's targets. Some pathways, I'm going to ignore that. And we see a totally different effect in aging. So the same phenomenon happens in that there's reprogramming, but it's different targets. And one of the main targets that we see in aged astrocytes that becomes rhythmic are autophagy genes. So this is mTOR. Everybody knows mTOR. It's involved in just about everything: aging, autophagy, metabolism. It's not really rhythmic in either wild type or APP mice, but then in aged mice, it's upregulated and it takes on this really beautiful rhythm. So it seems that somehow the clock is, taking over control of mTOR, and it's highly upregulated in astrocytes with age. Same with other autophagy genes like ULK1, ATG7. There's several others. Autophagy is on our list of pathways that seem to be change their circadian rhythmicity. So I essentially show this to just put it in the back of your mind. This will hopefully have this paper out soon, and it will have a website in which you can search through the entire transcriptome of, we've done astrocytes and we've done microglia at the same time, and we've done bulk tissues. So we have this huge data set that's going to be fully available, and we've finally made a website so you can search all this. So you can look for your favorite gene, if for nothing else, to make sure it's not rhythmic, in case you've been sacking your mice at different times of day. And but it's another element to think of when you're thinking of transcriptional regulation. Say, are these genes dysregulated? Or are they differentially expressed? Well, it may depend on what time of day you look. So the circadian clock, as I mentioned before, it can be manipulated, and we're sort of interested in what happens when you break the circadian clock to pathology. So if you knock out BMAL, that's the one gene in a mouse and probably in a human that completely abrogates circadian function. So the mouse goes from having nice wheel running rhythms to being completely arrhythmic when you knock out BMAL. We've shown that this causes astrogliosis, which I'll go into in a little more detail, really profound astrogliosis when you do this. and that's actually some more data here. So these are astrocytes in culture. They have these really nice rhythms in circadian clock gene expression, but if you knock down BMAL, you lose those rhythms. And the same thing happens in vivo. We've generated both full global knockouts of BMAL as well as astrocyte-specific knockouts, and these are all tamoxifen sensitive. So we treat the mice at 2 months old after they're fully matured. You cannot knock out clock genes in the germline very well. It causes a bunch of developmental problems. The mice have very different phenotypes, we've found. However, when we knock it out postnatally, you can see you knock out BMAL. This CAG is just a general promoter. It's like expressed in all cells. We give them tamoxifen at 2 months. We look at them 2 months later. You lose all the BMAL expression. This is CA1 of the hippocampus. There's a lot of BMAL in the neuronal cell bodies. It's all gone, and you can see the astrocytes look very angry. If you knock it out in astrocytes, you can see the BMAL in the neurons is retained, but the astrocytes, again, look really angry. And we have a paper in 2018 that goes into more detail about the cell autonomous regulation of astrocyte activation by BMAL. It's really, really potent. I've never seen so much astrocyte activation as in a BMAL knockout mouse. But the mice seem okay. They don't have microglial activation. They don't really have neurodegeneration. They just have a ton of astrogliosis, which is kind of an interesting phenotype, and I'll get into a little more about that in a second. So we were interested in what happens when we knock out BMAL in these astrocytes in disease models, because I'm not going to show it here. We have some previous data that when we knock out BMAL globally, it can cause oxidative stress in the brain. It can actually increase amyloid plaque formation in the brain if the mouse is arrhythmic. So we wanted to know what happens when we just knock it out in astrocytes, because when you knock it out in astrocytes, you get this astrogliosis without the mouse becoming arrhythmic. It's just only the astrocyte response. And so we used a couple different models to do this. This was recently published in Pat's paper in Neuron. One is an alpha-synuclein preformed fibril model, which was developed by Kevin Luck and Virginia Lee and published in Science a while back. You just make these fibrils in vitro. One of our collaborators, Gus Davis here at WashU, makes these fibrils for us, and we inject them into striatum. They cause misfolding of endogenous mouse synuclein that spreads all over the brain. And so we use this injection model in our astrocyte-specific BMAL knockout mouse. We give them tamoxifen a month before the synuclein, so they're already getting astrogliosis prior to the onset of the synuclein injection. And then they develop pathology for 3 months, and we look at it. And somewhat to our surprise, we found that normally you get a lot of phosphosynuclein, aggregated synuclein in these mice after 3 months in the control mice, but in the mice that are lacking BMAL in the astrocytes, you can see there's a little more green here. Green is GFAP, but there's a lot less red. This phosphosynuclein is reduced, and kind of everywhere that we look. I will say, if you wait longer, if you wait a year in these mice, we have some data suggest that this effect kind of goes away. So you may be able to overwhelm this response, but at least at 3 months, we see this pretty striking decrease, which is maybe not what you'd expect. And we think this is due to this astrocyte activation phenotype. And actually, I don't have the slide in here just for time's sake, but we see the same thing in a tau model. So P3ONS tau mice, when we knock out BMAL and astrocytes, they have much less aggregated tau than wild type or control pre-negative mice. So this same phenomenon holds true both for tau and for synuclein. So we've been very interested in why. What is BMAL doing in astrocytes that can somehow protect the brain from synuclein and tau? And so we did some RNA-seq experiments. Celia McKee is a former trainee who's now at Merck, did a lot of this work, isolated astrocytes from the brain using a Miltenyi kit, and then sequenced them. And we see a lot of genes that are dysregulated. But one of the main kind of themes that come up is endocytosis and lysosomes. So something having to do with protein degradation potentially. And when she looks more closely, she finds that in culture, when you knock down BMAL1 in astrocytes with siRNA, that when we use lysotracker, which measures low pH organelles that are oftentimes lysosomes, that there's more lysosomal content in these cells, which is quantified here. So just within a few days, these BMAL deficient cells can upregulate lysosomes. We see the same phenomenon when we look at LAMP1, which is another lysosomal marker, or RAB7, another lysosomal marker. And these cells actually take up and degrade various proteins more more efficiently than regular astrocytes in culture. And it doesn't matter what the protein is in our experience. It can be tau, it can be A-beta, it can be synuclein, it can be Celia just used BSA, like tagged BSA. So they take up and degrade proteins more. They become like little protein degradation factories. So Pat looked a little more carefully at like what some of the genes were that were dysregulated when you knock out B-mal and astrocytes. And I won't show all the transcriptomics, but one of the hits was this gene called BAG3, which is a known chaperone protein involved in autophagy. And Gail Johnson and others have shown that BAG3 is important for the clearance of tau in neurons. It's never really been discussed in astrocytes. But we see when we knock out B-mal, we get all these activated astrocytes, and they express tons of this BAG3 protein, specifically in the astrocytes. So we're very curious about this because it's involved in the exact process we were interested in, and clearly highly upregulated across different B-mal knockout models. And so we generated a virus, AAV GFAP promoter, either driving GFP or BAG3, and we inject mice at P0 intraventricularly, and then the virus kind of spreads throughout the brain and infects all the astrocytes. And we got pretty nice infection. You can see the astrocytes are turning green. They look like resting astrocytes. They're not particularly, they do express GFAP, but they have kind of a more of a resting looking morphology. But the ones that had the BAG3 construct express lots of BAG3. You can see it here. These are injected mice. The whole cortex is kind of filled with BAG3 positive astrocytes. And when we did our same model where we inject synuclein, you can see normally there's a lot of synuclein in the mice that were injected with the control virus. And in the BAG3 injected mice, there was considerably less. So we think just overexpressing BAG3 in astrocytes is enough to prevent phosphosynuclein, at least. We had some data from tau, but not quite as compelling. A little harder to work with tau than it is with synuclein. So BAG3, we thought, well, is this thing even expressed in humans? What's the deal with BAG3? It had only been described in neurons by essentially one pathologic paper. But usually the wealth of transcriptomic data sets that are out there in the world, the Seattle 80 Bray Atlas, our Knight ADRC, single cell data sets from our own place, and then all the other ADRCs from the US, you can see that in the astrocyte cluster here in this Seattle 80 Brain Atlas, it expresses GFAP, also expresses BAG3. So it seems like BAG3 is expressed highly in astrocytes in the human brain and in AD. It's upregulated in all of the major ADRC data sets that exist by about 1.5 fold. And when we look in astrocytes, so this is single cell data from patients with autosomal dominant Alzheimer's. So these are young people that have Alzheimer's disease. There's high levels of BAG3 kind of in these. You can see these clusters 2 and 4 only appear in the setting of disease. So we have these sort of disease-associated astrocytes. And in the paper, we argue they express a lot of the same markers, the disease-associated astrocytes that have been described in mice. And BAG3 is one of these kind of disease-associated proteins. So we think astrocytes are turning on BAG3 in the setting of disease, but maybe it's inadequate to prevent the damage that's already happened. So I don't know how I'm doing for time. Probably almost out. But I was going to how much time do I have? I don't seem to have like a timer here. 5 minutes?
About 5 minutes.
5 minutes, then time for questions, or 5 minutes total? I might just end it there.
Rebecca, what do you think?
It's up to you. You can have another 5 minutes and then 5 minutes of questions. We've got time.
Perfect.
All right, I'll do 2 more minutes. So really quickly, because I wanted to show some of Jian Li's work, we have some data that BAG3 is actually regulated by this other clock gene called reverb alpha that we've been studying for a while. So reverb alpha is controlled by B-mal. So when you knock out B-mal, reverb alpha levels go way, way down. Normally, it's a very highly oscillatory gene in the brain. This is in cortex. The gene name is NR1D1. But when you knock out B-mal, you basically lose reverb alpha expression. You get some astrogliosis, and you get high levels of BAG3. So We think BAG3 is downstream of reverb. Jian Li has been studying, she's a highly talented postdoc in my lab who will be looking for jobs soon. This is some unpublished data from her showing that if you knock out reverb in a mouse model, in the tau mouse model, the PS19 P3ONS mouse, that these mice, they have low levels of reverb, high levels of Cre. Again, it's a postnatal knockout. We give them tamoxifen at 2 months old. They have less tau. So if knocking out reverb will increase BAG3 and it causes them to have less tau. That's phospho-tau with AT8. Aggregated tau with MC1 is the same scenario. There's a lot in the regular PS19 mice, but in the PS19 mice lacking reverb, there's less. So it seems as though knocking out reverb suppresses tau aggregation in the brain. It reduces inflammatory markers. This is just qPCR, but it's GFAP, CD68, TNF-alpha, I1 beta, ApoE, Plin2 is a lipid marker. So it seems as though if you knock out reverb downstream, you can induce BAG3 and protect against tau. So this is important because you can target reverb with a drug. It's a receptor. It's actually a nuclear receptor. So there are drugs. There's actually a company that I collaborate with occasionally that's trying to make drugs to target this reverb nuclear receptor, potentially activate this pathway in vivo. But it's not always so simple. This will be the last thing I'll talk about. So Jian recently published a Nature Communications paper where we knocked out reverb only in microglia. So I haven't talked about microglia at all, but they have a whole different scenario. So it turns out when you knock out reverb in microglia, the male mice only develop more tau pathology, and they're more inflammatory. So if you give them LPS, they have more responses. It's only male mice. So microglia seem to have a lot more profound sex differences in their responses. And I know many of you have probably seen that in your work. But these mice that have microglial reverb deletion, in the male ones at least, they have more inflammation, they have more GFAP, more IBA1, more tau across multiple models. So this is very cell type dependent, context dependent, what the clock is doing or what clock genes are doing. And we think this is because reverb seems to regulate microglial metabolism. So lipid metabolism is one of the hits that came up when we sequenced these reverb deficient microglia, and they collect lipid droplets very easily in culture. So cultured reverb knockout microglia just have spontaneous lipid droplet formation that you can see quantified here, higher levels of Plin2, which is a protein that marks lipid droplets. And Jian did some work showing that when you induce lipid droplets with oleic acid, you can induce lipid droplets and you can block it with a DGAT1 inhibitor, that this actually suppresses phagocytosis of tau by microglia. So microglia that have a lot of lipid droplets, and Tony Weiss-Correa's lab and others have done work on this before we did, but she shows that if you induce lipid droplets, you can suppress tau uptake by microglia really profoundly. You can only rescue this by a little bit, although by blocking lipid droplet formation with DGAT1. So there's something else going on with this lipid overload in the cells. We see a similar phenomenon in reverb knockout cells. They have less tau uptake, and you can only partially rescue that with a lipid droplet inhibitor. So we're very interested in sort of immunometabolism of microglia and how the clock is regulating that, but it may kind of fly in the face of what we see when we knock it out globally. So the clock has very different functions in different cell types. So we just have the tip of the iceberg here. We know the clock's important for regulating a lot of genes involved in neuroinflammation and disease process, but exactly how that works and exactly what the context is and what we should target therapeutically is still up for debate because it's a fairly complex system. So just think about time of day when you're sacking your mice, when you're doing your experiments. I'd be happy to answer a couple questions with whatever time I have left.
Erik, that was amazing. Thank you so much. I've been here scribbling little notes to myself because I work on microglia and metabolism, and I'm actually delighted to hear that you're going to be putting this resource online. It's so important. These change in the genes, just the different times of day. I think that's going to have a really big impact. Because as you know, I mean, big mouse culls, you don't often have a choice what time of day you're sacrificing different groups and so on. So this is really, really interesting. Thank you so much.
Sure.
A quick thought that popped into my mind was, have you tried single cell sequencing or anything like that? Or do you know if it's the plaque adjacent astrocytes that are driving these changes, or global astrocyte populations, perhaps?
Yeah, that's a great question. We don't know that yet.
Okay.
Because this is just grinding up. You need quite a bit of tissue to sort of get enough. We have
Absolutely.
tried single cell. It is difficult because these oscillations are not huge. They're maybe like 50%. Some of them are only 30, 20%.
Okay.
Single cell is kind of noisy, and we just have not been able to get, I mean, if you did billions of cells, you might be able to see something, but we could see changes in a few cells, but like we might get like a handful of genes, I'm sorry, a handful of genes that are oscillating in a given cell cluster.
Yeah.
I don't think we're quite there yet. I know people are working on it, and so hopefully we'll be able to move into that. I think spatial transcriptomics has a lot of potential there too. Like we would like to do that, to look around a plaque to see, can we see an oscillation in a given gene?
Yes.
But I think that the beauty of the bulk sequencing involved with TRAP and ribotag is that it gives you just such clean, really, really clean data that you can actually see a 20% change. I mean, you can see even like a 5% change. And the clock is amazing. You could just take mice, randomly kill them at different times of day, and like put all the data together, and they clearly show this beautiful, it's very robust.
It's very striking. It's really striking. We actually have a few questions now in the chat. I'll just read them out. Lots of people saying great talk and thank you for the amazing talk. Do you know if you disrupt sleep in mice, whether that also changes the circadian rhythm of the clock genes? And if so, would the APP-PS1 mice perhaps respond in a different manner or similar manner with the different clock genes?
Yeah, it's a great question. So sleep and circadian rhythms are just intertwined. It's very hard to separate them entirely. So certainly knocking out clock genes influences sleep. So in humans, a lot of the sleep phenotypes that we see are driven by clock gene mutations, actually. The other way around is a little less clear. Certainly, if you fully sleep deprive mice for a night, you do see some changes, modest changes in clock gene expression. We don't know what happens in a disease model. So, my mentor, Dave Holtzman, does the sleep stuff, so I try to avoid it some, but we are doing some of those experiments to see if we sleep deprive a mouse and we did this exact same thing and look at astrocyte transcriptome, like what happens. We don't know that. And it could be that there's an interaction between plaques and sleep deprivation.
Yeah.
There's a woman in Dave Holtzman's lab, Samira Parzakar, who's just published some papers and has another paper coming out about this kind of work that's really pretty cool about how sleep deprivation also affects microglia.
I've seen some of the work. I would be really curious if you could merge his model with your model. I wonder how the mice themselves would, it's so much to tease out.
Yeah, that's a good idea. We have a grant just like that. It didn't get funded. So. Also, I'll suggest you as a reviewer next time.
Anytime. We have another question here. Let me see. A really great talk. They liked the part that you used TRAP and that she also used the same approach like ribotag. Is it a global TRAP profiling or specific brain regions? And have you used the AAV TRAP to target specific regions? And this is a question from Boston.
It's like the whole cortex, essentially. So we have not tried to target it beyond that. But yeah, we have not yet used the AAV TRAP, but that's a really great tool. We just used a mouse, ALDH1L1 RPL10A mouse. So yeah, there's viral versions of this where you could just inject into, say, the striatum or the substantia nigra or something. We have not done that, but that would be really cool to do.
That's really interesting. And with the change in the astrogliosis in the BMAL knockout, have you ever checked if GFAP levels in the plasma increase? Because GFP, as you know, is often used in clinical trials as a biomarker.
No, we have not done that. That's an interesting question because, yeah, GFAP is becoming very popular for AD. There are other, so we have a paper about Chi301 YKL40, which is another Alzheimer's kind of biomarker that is regulated by the clock and actually becomes rhythmic in the setting of plaques. But we have not looked at GFAP in the blood of these mice. I would assume it'd be elevated, but if it's not, it'd be pretty interesting.
Do you actually see changes in the blood-brain barrier with your BMAL knockout?
It's an interesting question. We do not, just under basal conditions. However, I have a grad student working on this, that when you add an inflammatory, if you give them inflammatory, like LPS at different times of day, there's a very different response to the
super interesting.
blood-brain barrier integrity. So it's a little more complex.
Yeah, we have a question similar to that on whether knocking out some of the clock genes impacts the 24-hour cortisol rhythm as well.
Absolutely. So if you disrupt the SCN, I didn't kind of go into detail on the whole clock system, but the suprachiasmatic nucleus is like the central clock of the whole body. It's highly connected to cortisol rhythms. So if you disrupt the SCN, you will lose cortisol rhythms in the animal. Now, in the astrocyte-specific B-mel knockouts, we haven't seen any difference in cortisol. So you really have to disrupt kind of the central clock to see that. But cortisol, there's two things that are always like in the back of my mind. How's this affecting sleep, and how's this affecting cortisol? And the audience is very sharp to like notice both of those. Those are both so important to like every aspect of our function that, you know, these things are all sort of intertwined. But you can get at it with some, you know, tissue-specific knockouts. And I don't think a cortisol change is affecting at least the astrogliosis response we see, because we even see it in culture. I didn't show that, but in culture you can see it too.
Yeah, and I thought that was very interesting what you showed at the beginning, that you can track these oscillations in cell culture. Really remarkable. Perhaps one more question here. They were wondering about the B-mel changes on epigenetic marks or the oscillatory expression of the respective transcription factors. Have you had an opportunity to look in that direction, perhaps?
We have not. There's a woman named Paula Desplatz at UCSD that's doing a lot of epigenetics. She just had a really cool cell metabolism paper about time-restricted feeding in APP models that seems to be protective. She has all kinds of epigenetic data in the brain showing that there are rhythms in epigenetics. She's published a prior paper on this topic. So the clock and epigenetic regulation are very closely connected. And actually there's some data that suggests that clock, like the protein clock, is a histone acetyltransferase, so itself, which could like directly link these two things. So my guess is a lot of the reprogramming is epigenetic. And actually there's some data to that effect in other cell types from Paulo Sassoni Corsi's lab. He was sort of like, you know, he's passed away now, but he was kind of the forefather of this sort of, you know, line of inquiry. And their lab showed some epigenetic changes in liver, particularly.
Okay. Erik, this was wonderful. Thank you so much.
Hey, thank you all.
I really enjoyed it. And now, just due to time reasons, we'll move on to our next speaker. So now we have our early career research speakers. So these talks will be a little bit shorter. And we'll start off with Jacob Dundee from the University of Cambridge in the UK. So Jake originally studied for his undergraduate degree in biochemistry at the University of Bath. He then started his PhD at the University of Cambridge, working on microglial phagocytosis in a variety of contexts under Professor Guy Brown. During his time in Cambridge, he has co-created a website collating microglial research, microglia.info, and he has hosted the European Microglial Webinar Series. He's organized a conference with the Biochemical Society and Microglia and Neuroinflammation in addition. Jake is less than a month from submitting his thesis and will shortly start a postdoc at Baylor College of Medicine in Houston, Texas. So Jake will talk to us today on the role of the P2Y6 receptor in aging-associated synapse and memory loss. And again, please don't forget to add your questions into the question box. Thank you so much.
Thank you so much. And thank you so much, Proteintech, for inviting me to speak here today. I really appreciate it. I love talking about microglia, so it's always a great chance to do it. So what I'm going to talk to you guys today about is about the P2Y6 receptor, specifically in the context of aging-associated synapse and memory loss. We'll kind of real focus on microglia. So first we'll kind of give a wee summary into microglia. We just had a lot of things about astrocytes and we touches and drabbles about microglia. But microglia are central nervous system resident macrophages, and they're involved in a lot of different things. They're really important for your brain. So like the macrophages in your blood, they carry a lot of different functional roles in maintaining a healthy brain, whether that be surveying the area in the brain or releasing cytokines to kind of regulate the inflammatory or the inflammation of the brain. One particular aspect that, particularly in Guy Brown's lab, that we focus on is this function of phagocytosis, or eating of different things, whether that be an aggregated protein like in Alzheimer's disease, or a synapse, intention like development as well, and help form, ensure that you have the correct number of synapses there, so you have a really efficient neural network. So they're very important for a lot of things. So what we've done in our lab, in Guy's lab, is generally try and characterize phagocytosis and try and understand what signals do microglia use to determine whether to phagocytose something or whether not to phagocytose something or to eat something. And we kind of think about it as a big phagocytic code, right? Where you have a variety of different signals that either encourage phagocytosis or stop phagocytosis. And one of the most common ones being phosphatidylserine, this sort of apoptotic marker or stress marker that's flipped onto the surface, which counts as an eat me signal, which is then typically bound to by an opsonin such as Gas6, and then which then kind of bridges the gap between phosphatidylserine, the eat me signal, and the phagocytic receptor on the surface of microglia such as MERTK or TREM2. So we think it's really important to sort of characterize these different receptors, especially because phagocytosis as a whole, it's very broad, right? There's a lot of different things that's really, really essential for microglia to phagocytose. So we don't actually want to, if, thinking therapeutically, especially in terms of aging, you have this sort of general prolonged neuroinflammation or this prolonged microglial activation where microglia might be phagocytosing more than they would do in adult life or early childhood. We want to be able to stop phagocytosis of things that are detrimental to the brain, but still maintain the phagocytosis of targets that are beneficial to the brain, such as the clearance of aggregated protein. So characterizing this phagocytic signal better, gets us a better understanding of what is a good sort of marker or phagocytic line to target therapeutically. And in particular, I work on the P2Y6 receptor. But what exactly is the P2Y6 receptor? Well, it's it's a G protein coupled receptor that's found primarily in the brain on the surface of microglia, and with a small subset of neurons that also contain it. And with its main ligand being UDP and kind of similar to a lot of G protein coupled receptors. Once ligand binds, it induces PLC-induced calcium rise. But why do we actually care about this receptor? Well, not only is it microglia being the main source of it in the brain, actually it seems to be very important in phagocytosis. So this is back in 2007, there's a research called Kazumi in Inui's lab back in Japan. They found that if you take primary microglia from rats, and then you feed them these fluorescent zymosan particles, and then you add UDP, being that ligand for P2Y6, you see about a 10-fold increase in phagocytosis, suggesting that P2Y6 is the activation of P2Y6 induces the phagocytosis or induces phagocytosis in microglia. And not only that, if you stress out neurons from rats and with canic acid, which is an excitatory stress, it actually induces the release of UTP, UTP being urine triphosphate, which is then thought to be quickly hydrolyzed into UDP, that P2Y6 ligand. So this was done in vitro as well as in vivo. This is doing like a little dialysis system to look at the UTP release. So that's suggesting there that actually neuronal stress, at least an excitatory stress, is inducing the ligand, the release of the ligand for P2Y6. That's interesting. So Guy's lab kind of took this on a bit further back in 2014, so a couple of years later, and they wanted to investigate P2Y6 more, especially in the context of neuronal loss and sort of inflammation as well, and see if there's any sort of important link to that. So to do this, they would isolate microglia neurons and astrocytes from mice and to this sort of tri-culture system, and they're called CGC cultures or cerebrogranular cell cultures. And they're useful for a lot of really great things, especially sort of a 2D sort of modeling of inflammation. But in this situation here right now, they showed that actually if you grow these cultures up and you add UDP, it induces a significant amount of neuronal loss. So the ligand to P2Y6 is inducing neuronal loss, and better to confirm it as well is by adding MRS2578 being a P2Y6 inhibitor, which then completely prevented the neuronal loss observed by adding the P2Y6 ligand, showing that P2Y6 induced neuronal loss, or P2Y6 is inducing neuronal loss, yeah. And on top of that, if you add LME, which is a compound which you can use to deplete microglia from these cultures, you could then don't see, if you add it with the UDP, you don't see neuronal loss, showing that actually the neuronal loss induced by UDP is both P2Y6 dependent and microglia dependent. This is then further verified or further investigated again using the same CGC cultures except from wild type and P2Y6 knockout mice. And you can see the UDP does induce neuronal loss and which is then prevented in the P2Y6 knockout mice. But then obviously it's all well and good adding UDP to neurons and watching them die. But obviously in the brain, we're not, it's not as necessarily as common, or it's not as, the neurons might be releasing it, but in really small doses. So it might be more useful to use maybe a more representative model, or more representative of maybe what's happening in an aging brain, for example. So then what I also looked at is using LPS. So I believe Erik talked about this a little bit. LPS is lipopolysaccharide found on the surface of gram-negative bacteria, and we often, scientists, use it as sort of an inflammatory or neuroinflammatory stress, as a way to sort of sometimes activate our cells. And you can do this in CGC cultures, and you can add LPS, and it actually induces a microglial-dependent loss of neurons, specifically through phagocytosis. So by adding LPS, you do see neuron loss, and actually by blocking P2Y6 again with MRS2578, you actually see a substantial reduction, a significant reduction in that neuron loss induced by LPS. So suggesting that the P2Y6 is potentially involved in this sort of inflammatory induced neuronal loss. That's further confirmed as well using P2Y6 wild type and P2Y6 knockout mice as well. Finally, switching gears a bit, they're now looking at sort of an in vivo model, looking in the context of neurodegeneration instead, sort of aging or sort of inflammatory. This is using P3RNS mice, slightly different to the PS19 mice Erik talked about previously. And this is a model developed by Maria Ciantini and Michelle Goddard, where actually you can they do substantial amounts of, about 6 or 7 months old, you see a substantial amount of tauopathy in the brain, as well as a substantial amount of memory loss. This is by the novel object recognition test. You see a substantial amount of memory loss, with only a really small amount of neuronal loss that seemed to cause that amount of memory loss. But if you cross these with P2Y6 knockout mice, you see complete protection from memory, and a non-significant, a slight reduction in neuronal losses in regions like the perihinal cortex. Which is really interesting. So that's suggesting, well, actually, okay, you're seeing neuronal loss here in this neurodegenerative model, but not a very substantial amount. And that amount is seeming to be able to cause such a large change in memory, suggesting that maybe neurons isn't the big thing that's changing in this situation, suggesting that maybe there's something else, maybe some other subcellular structure of neurons that could be affected instead, that could be tied more closely to memory loss in this situation. But then obviously keeping the context of Alzheimer's disease, I think this graph many people who kind of look at Alzheimer's disease recognize in some degree, where the amyloid plaques, we kind of focus a lot of research into amyloid plaques, but in general, amyloid plaques don't generally correlate as well with the symptomatic onset of Alzheimer's disease, that memory loss that we all sort of remember, that we all kind of know Alzheimer's disease for. They don't correlate particularly well with it, and tau tangles do slightly better. But actually what seems to correlate much better with memory loss is actually sort of the decrease in brain structure, or that loss of like neurons or synapses, the connections between neurons. That seems to be kind of correlate much better with memory loss in sort of Alzheimer's disease. And actually similarly in aging context in humans, during aging, about 40% of people over the age of 65 will experience some form of memory loss in their life. But actually there's no significant change in neurons or decrease in neurons during that time period. But what does seem to significantly change is the number of synapses that are there, suggesting that actually we should probably, especially if we're looking at this sort of symptom onset in aging and neurodegeneration, we should look at synapses a bit more closely to see what's going on there. So with all this in mind, I've thrown a lot of information at you guys. With all that in mind, what the general idea about P2Y6 right now, or what we think about what is happening for P2Y6, is where you have neuron, and a neuron might undergo some sort of stress, whether that be due to the collection of neurofibrillary tangles or tau tangles inside, or an external stress, whether due to damage due to an infection or amyloid beta plaques, or sort of a local damage elsewhere that's causing neuroinflammation or neuroinflammatory damage to the neuron or stress to the neuron, which results in the release of UDP or UTP, which then activates P2Y6 and by local microglia, inducing the phagocytosis of that neuron or other subcellular targets, such as like a synapse or a myelin. So with all this in mind, I wanted to investigate this a bit further in the context of aging, because we've shown already that actually P2Y6 can knocking out P2Y6 can protect against neurodegenerative induced memory loss. But is it similar to aging, especially because there is this link with aging induced memory loss in synapses? So to do this, kind of do this two kind of avenues. The first being through an in vitro work, similar to the sort of CGC cultures I talked about before, the triculture system with neurons, astrocytes, and microglia, as well as just using microglia by themselves. These are isolated from the cortex or the cerebellum of both wild type and P2Y6 knockout mice, and sort of tease out the sort of mechanisms behind what are going on here. And then also looking in an aging context, looking at 4 and 17-month-old P2Y6 wild type and knockout mice, and then looking structurally, do the synapse levels, do any other sort of pathological changes of aging seem to occur? Are there any changes in P2Y6 knockout mice? So I start off by seeding some microglia, some primary microglia, with these sort of fluorescent beads, these 3-micron beads that have on the surface phosphatidylserine, being that sort of apoptotic eat me signal I mentioned earlier. This isn't a perfect marker, but it is a sort of a marker of an apoptotic cell. And you can treat these microglia, wait a certain amount of time, and then measure their sort of uptake by flow cytometry and by looking for the amount of green fluorescence inside them after a certain amount of time. And if you look at P2Y6 wild type and knockout microglia, you actually see no change in the sort of uptake on that, which is what we kind of expect, right? If you imagine P2Y6 is not being activated or involved, then it's unlikely it's going to be changing the phagocytosis. But it's important still to look to see if actually P2Y6 deficiency would affect phagocytosis of microglia. Is that signaling so important that actually microglia lose this function because of it? And it doesn't seem like that changes it. But then looking at maybe a more physiologically relevant target, instead, what we could do now is isolate synapses called synaptosomes from a rodent brain, and you can label them with a substance called pH-rhodo. What this substance basically, what this dye basically does is it will fluoresce red when it's in an acidic environment. So once a microglia has phagocytosed something that's pH-rhodo labeled, it will then be trafficked to the lysosome, which will then induce a red fluorescence, showing that microglia has phagocytosed the target. And you can do is you can treat these wild type and knockout microglia with synaptosomes, and actually you see a significant reduction in the amount of uptake of these pH-rhodo labeled synaptosomes in the knockout mice, suggesting that P2Y6 deficiency or P2Y6 is involved in the phagocytosis of synapses. Now that's all well and good. That's an in vitro model, and it's just adding these kind of mushed up synapses to microglia by themselves. Let's take it one step further. So now looking in these CGC cultures again, this triculture system, and actually, like before, I said you can add LPS to induce neuron loss, but you can add slightly less LPS and just induce synaptophysin staining loss. And if you do that, you see in the wild type mice, you see a clear reduction in synaptophysin in staining. But if you then look at the knockout mice, you don't see any LPS-induced synaptophysin loss. Now, clearly, I must point out here, there is a reduction. There seems to be a non-significant reduction in synaptophysin staining in the knockout mice. However, the LPS-induced synaptophysin staining loss does not seem to change, further supporting that maybe P2Y6 is involved in the phagocytosis of synapses in particular. So then to look at this further, I looked in 2 different regions in these sort of young and old wild type and knockout mouse brains, and I wanted to look at the sort of some key regions involved with memory, such as the hippocampus, I looked at the CA1 striatum varietum, as well as the somatosensory cortex, and look for synapses by looking for the co-localization of a pre and a post-synaptic marker. In this case, the pre-synaptic marker being VGLUT1, and the post-synaptic marker being HOMA1. This is for excitatory synapses. So you can stain brains for these 2 different markers, and then by using confocal microscopy and taking a Z-stack, you can then basically investigate or look for puncta of a pre-synaptic marker and a post-synaptic marker. And if they're within 200 nanometers of each other, we would classify that as a synapse, suggesting that they are connected to each other through that. And if you do this, you see in both the somatosensory cortex and the hippocampus, you see a significant reduction, an aging-induced reduction in synaptic density. in wild-type mice, which is to be expected. However, if you look at the P2Y6 knockout mice, you don't see a significant aging-induced reduction in synapses, which is really interesting, suggesting that potentially P2Y6 is involved in aging-induced synapse loss. Again, I will point out, if you look at the somatosensory cortex, especially in the young mice, you do see a slight reduction. It was non-significant, but a slight reduction in the synaptic density in the young mice compared to in the knockout mice compared to the wild-type mice. Suggesting that maybe something else is going on there. And P2Y6, and actually, because these mice never had P2Y6 to begin with, suggesting that potentially P2Y6 is involved in the development of synapses at some point. Finally, well, not finally, there's a little bit more. But anyway, there's the next one to look at is we've shown sort of phagocytosis in sort of an in vitro model, but now we wanted to investigate actually in vivo, are we seeing this sort of increase in or changes in phagocytosis in this context as well? So to do this, it was a great methods paper by Dorothy Schaefer, looking at the internalization of synaptic material using sort of confocal microscopy and this program called IMRES. And by using, you can stain microglia for IVA1 and then stain their lysosomes using CD68. And then you can look for the internalization of synaptic material, in this case, VGLUT1, that presynaptic marker, within the lysosomes of these microglia. You can do check a Z-stack of this and then look, do a surface rendering and look for the internalization of that. It's very pretty. I never get all of these images. And if you do that, you see, first of all, you see an age-induced increase in CD68 volume, regardless of genotype. This is relatively expected. This is what we kind of expected, as we highly reported to kind of occur during aging. And there's a lot of different reasons to why this might be occurring. One being sort of this idea of increased neuroinflammation in general in the brain, inducing greater microglia activation or phagocytosis because of it. But then actually, if you look inside the lysosomes, in aging, you see a significant increase in VGLUT1 internalization in the P2Y6 wild-type mice. But in the knockout mice, you don't see that at all. You see no aging-induced increase, and it's quite a stark, increase in the wild-type mice. Further suggesting that P2Y6 is involved in the aging-associated aging-associated synapse loss that occurs through microglial phagocytosis. Finally, to kind of bring it all home, doing just a novel object recognition test and to test these aging mice's memory. It's well reported that aging mice around this sort of time period do experience memory loss, and as you can clearly see, the way this sort of test works is you'll put a mouse into a room, you show it two objects, and you'll change an object. And if they spend more time looking at the new object, they remember the object. And you see an aging-associated memory loss in the wild-type mice, which you don't see in the knockout mice. But once again, I will point out, there does seem to be a slight non-significant decrease in memory, or at least how they perform in those tests in the knockout mice, the young knockout mice versus the wild-type young mice, which is interesting. So to conclude, all this sort of information overload I've given you all, it seems that P2Y6 influences microglial phagocytosis of synapses, and that actually P2Y6 is involved in the aging-associated synapse loss through microglial phagocytosis of those synapses. And then that loss of synapses actually relates to the aging-associated memory loss we see in these mice. Obviously, I mentioned some points throughout, that the young mice or the P2Y6 knockout mice at some point seem to have reduced synaptic numbers and potentially reduced synaptophysin staining, suggesting that maybe P2Y6 might be involved in development as well, and potentially affecting synapse. So this idea of synaptic pruning in like forming those neuronal networks. I don't have time to talk about today, but if you want to read more, feel free to. Thank you again for listening, and thank you so much for Proteintech for giving me a chance to talk.
Thank you, Jake. That was really interesting. Lovely overview to your work. I was curious if you, because of course this is just a short talk, but did you have a chance to characterize your P2 or Y6 knockouts? So do they increase any other phagocytosis receptors, or do they have any functional changes like microglial motility changes, perhaps?
So I investigated, that's a really, really great question. I investigated a couple of different things in vitro. For example, I did migration, for example. I did obviously just using a simple transwell assay. I didn't see any sort of changes to sort of general motility. I also did this cytokine array as well, and I saw a few chemokine changes, but the sort of key, inflammatory cytokines in response to LPS, such as IL-6, IL-1 beta, your TNF alpha, were all kind of the same, and the changes were very few and far between. So on a whole, they seem pretty similar.
Pretty similar.
Mm-hmm.
And we have a question here as well. So what are the drug discovery implications of P2Y6 receptor knockout? So it seems they can promote phagocytosis, but agonists could also lead to neuronal loss and promote inflammation.
That's right. So yeah, I guess in the context of my research, there's definitely a push to potentially inhibit the P2Y6 receptor to try and prevent, especially in the sort of neurodegenerative or the sort of the aging context, and try and preserve the synapse, or stop synapse loss and preserve memory. As best as though I do know some people who are looking at P2Y6 agonists as well. Like I said, in the brain, P2Y6 is predominantly on microglia. There's not many other cells that have P2Y6 in the brain, but throughout the rest of the body, there are other cells that have P2Y6. So it's important to sort of have those considerations as well if we're talking about drug targets.
Yes, absolutely. And accessing the brain is so challenging in itself.
Absolutely.
Okay, well, due to time reasons, we'll just move on. But thank you so much for your really interesting talk.
Thank you so much.
Now we have Dr. Danielle Beckman from the University of California. University of California, Davis from the US. So Danielle Beckman is an associate project scientist at the California National Primate Research Center from the University of California. She has a master's degree in biophysics and a PhD in biochemical chemistry. She has been working with primate research over the last 7 years with a primary focus in proteinopathies and mechanisms associated with neurodegenerative diseases such as Alzheimer's disease and related disorders. And today Danielle is going to speak to us about developing a novel monkey model of Alzheimer's disease. Thank you so much.
Thank you. Thank you, Martin, for the introduction. Thank you, Rebecca and Proteintech for the invitation. Let me just share my screen really quick. Okay. I hope it's showing. Okay, so I'm going to talk a little bit today. Let me just see the time to make sure I don't go over. Okay. I'm going to talk a little bit today about, I tried to put some of the data, recent data we have with our COVID monkey model, but I'm mostly going to talk a little bit about our Alzheimer monkey model that we have been developing. And yeah, so I'm excited because there is a lot of microglia, astrocytes, or a lot of glia talks today. So mine is going to be another one. I'd just like to do an introduction about the primate centers, because a lot of people are not aware that what exactly we are and where we are. So there are 7, at least in the US, there are 7 primate center, national primate centers across the country. You can see where each one of them is located here. And our primate center is in Davis, in California. It's close to Sacramento, which is the capital. And here in our primate center, we have a very, very large colony. So yeah, you can see where we are here in Davis, Sacramento, in the central area of California. We have a very large colony. We have approximately now 4,500 monkeys. You can see the 2 most common species we have is the rhesus macaques. You can see them eating pumpkins because we received some donations of pumpkins because of the Halloween that just happened. So they enjoy a lot getting some pumpkins. We have also the TT monkeys, which are models for monogamy. They have high oxytocin production. And the thing that we are very proud is that we do breed our monkeys here. And we also we have been breeding them for more than 20 years now locally. And we have like 9 generations. So we have been behavioring, characterizing them, and also have, besides the other studies, these monkeys are engaged. And I think one of the things we are very, very proud is that we have an aged colony. So we're trying to increase, and we have now some money from the National Institute of Aging for keeping these animals. So we have around 100 animals that are 15 to 19 years old. And we have about 90 animals that are 19 years or older. And remembering that these animals, that's about 3 times the rate of the human aging. So this is very, very interesting because we are able to see much faster than using humans. We are able to follow like age-related cognitive decline and also age-related diseases. So these monkeys are used for different studies. in our primate center, but also like we are very much looking forward to collaborate with other groups. So if you're interested in doing some type of research with these aged monkeys, please reach out. Talking a little bit about our Alzheimer monkey model. So this is something that we started to do recently, this tau monkey model. So we published this paper in 2021 that we're doing just this initial characterization of the model. So we are looking at the 3 months. So what we do is we inject a double tau mutation, so the P31-1L and the S320F. So it's common mutations that are found in that are used to do mice models for tau pathology, to observe tau pathology. We injected them in the entorhinal cortex of the rhesus macaques, so they are like young adults, rhesus macaques. And in this study, we look at the brain of these animals 3 months after infection, so after the injection in the entorhinal. So as you can see here, and we try to classify, we do a lot of microscopy, and we were trying to classify, try to see the temporal progression of the tau pathology in the neurons. So we can consider like a pre-tangle when we see 88, which is a misfolded tau marker that is very commonly used. When we see thioflavin, which is a marker for more mature fibers, we consider them mature tangles. And when we don't see new end staining anymore, so like the neuron is really dead, and it's just, you see just the thioflavin mostly, just like accumulating in the neurons without any other marker, is usually ghost tangles. And what we saw in the 3 months, and looking at the entorhinal, which is the injection area, we saw some of this like mature tangles, but a lot of pre-tangles, and mostly pre-tangles, but some of the mature ghost tangles, especially in layer 2. But in the hippocampus, we saw a lot of pre-tangles, mostly in very, very few ghost tangles and mature tangles. So this was 3 months after the injection. This is what we show like 2 years ago. And what we are doing now, and this is like study that it's under review, is like looking at 6 months after the injection, right? Because 3 months is very early, it's very fast. So interestingly, what we saw when we waited a little longer, so instead of doing the same injection, but waiting 6 months after the injection, we saw that there is a lot of ghost tangles and mature tangles. You can see like it's mostly, you don't have pre-tangles anymore, and you have a lot of like, even the layers got very thin, so you have a lot of neuronal loss in these regions. So not only in the injection site, but also in the hippocampus. And I think the most interesting thing for me is the inflammatory component of these diseases. So what we see is that in the 3 months time point in the CA1 and the hippocampus, we see a lot of glia and astrocytes activated, but at 6 months, this is like very, very high. So we do see a lot of microglia directly interacting with neurons that are like producing misfolded tau. And you can see here that even some of these neurons that are completely engulfed by microglia. So as shown in this video, so this is something that it was like very interesting for me, because I'm like very, very interested in how these neurons containing misfolded tau can interact directly with microglia and astrocytes. So because we have 2 time points now, and actually we are also 3 and 6 months, but we're also looking at 6 weeks now. But what we were trying to do is just using a combination of different tau markers for misfolded tau pathology. So looking for like transsynaptic to finally ending at the late stages of pathology. So we are able to see, it's very rare to be able to see this in the human brain, because usually when we have access to the human brain is already later in the pathology. So because we're able to see like these different time points in the monkey brain, we're able to characterize different markers for misfolded tau. So we go from tau oligomers to like early events, like early truncation, not only phosphorylation, then you have phosphorylation of different sites, you have late truncation, and finally we have fibrillary tangles. So you can see the morphology of the neuron is very different across these different epitopes. You see very much the complete neuronal structure early, and it's gonna get lost. So you lose the dendrites, you lose axons. So at the end of the disease is like these neurons don't have neuronal markers anymore, so they're mostly dead, and you just see like the ghost tangles. And for me, what is the most interesting thing is that we saw that these different stages of pathology, microglia and astrocytes, are interacting differently with these markers. So we see that early in the disease, there is a lot of microglia interacting with these early misfolded tau markers. So tau oligomers is very much, the neurons containing tau oligomers are very much interacting with the microglia in green. And when we go progressing in the disease, we see like tau truncation, phosphorylation, the serine 42, which is also early terminal exposed. So these are very much interaction with microglia. But later in the disease, mostly starting with the 88 and PHF1, we see that mostly the microglia is not so much important. It's much more like astrocytes are interacting, and we see a lot of microglia dying locally too. So we think that the microglia is getting overwhelmed, maybe with the amount of pathology and an amount of neurons dying. And the astrocytes are mostly the ones responsible to clean the mess. Let's put it like that. So in summary, what we're seeing is just that we see this progression of early to late tau pathology transitioning from early tau phosphorylation to mature tangles. We see a difference in the microglia astrocyte crosstalk in the brain of these animals. So we see a lot of like normal microglia and astrocyte in the beginning, then you see a lot of microglia activation, then shifting to astrocyte activation later on. And we see a lot of neuronal loss, not so much at 3 months, but much more at 6 months. I didn't have time to show, but we also see this in the MRI. So we see hippocampal atrophy and also in PET scan, marking for tau. So we see very localized tau pathology and hippocampal atrophy in the injected sites of the brain. I also want to talk a little bit very fast about the other models we have in our lab. So we are also working with 3 different types of models that are like linked to the viral infections. So these 3 models were also published last year. We have a model with like we're trying to do HIV associated neurocognitive disorder so we are looking at the aged monkeys infected with HIV and looking at their brains. We have a model for COVID-19 that we are also very much interested how the infection with SARS-CoV can impact, especially the aged brains. And we are also looking at diabetes component in these models. And we have a model of Zika infection that is a neonatal, but is also like looking at inflammation. And the biggest link between all these models is neuroinflammation. So how microglia and astrocytes are like taking care of the viral presence, different types of virus in the brain. And very briefly, I just want to show a little bit of the COVID model that we recently developed and we published last year. But the interesting thing is that we are looking at very, very early infection. So just 7 days after infection, we're doing a lot of microscopy and different analysis. And we are doing 4 different groups. But the interesting ones are the aged animals. So we have aged infected and non-infected animals. And it's very interesting for us because we saw a lot of difference. We see a lot of difference in the young versus aged animals. So we see way more pathology. So as you can see here, we are staining for neurons, MAP2. We see viral double-strand RNA concentrated in the neurons mostly. And we see a lot of spike protein in these neurons as well. And we saw that the microglia and astrocytes are very much reactive around these neurons that are infected. So you can see here, so when we are looking at diabetes, we are also looking at diabetes because age and diabetes are 2 major components in COVID-19 infection. So we are also seeing if there is this component in monkeys as well. We have monkeys that naturally develop diabetes, same as humans. So using that, we were able to see, okay, how is the infection? It gets worse with aging, gets worse when the animals are that have diabetes. And we saw very much an increase in inflammation, like shown by much more presence of reactive microglia in the olfactory pathway of these animals. I don't have a lot of time to show, but This was published last year. So it's basically, we see the interesting thing is that looks like at 7 days after infection, the virus is spreading faster in the brain of aged animals in compared to young animals. We see a lot of the virus in the primary and the secondary olfactory cortex, and even in the tertiary olfactory cortex of aged animals, but not in young animals. So it looks like some types of, like the brain of the aged animals are more vulnerable to the viral spread. And of course, like looking very much at the neuroinflammatory component. So how much of the inflammation, how much we see a lot of the microglia mostly reacting and interacting with these neurons that are infected. And we see very much a big difference between the aged controls, so the animals that are not infected versus the infected animals. We're using super resolution air scan to be able to detect that. And the last part I want to show is just something we are doing right now. So one of the interesting things about these aged monkeys is that they accumulate and they produce amyloid plaques, the same as humans with age. So not necessarily is Alzheimer, but they produce amyloid plaques. And we see a lot of microglia and astrocyte activation around these plaques, and also here standing for amyloid oligomers, so showing that there is a lot of astrocytic and microgliosis in response to amyloid presence. So exactly like humans. Well, what we are seeing when we infect these very old animals that have amyloid plaques with SARS-CoV is that there is a colocalization between the presence of beta amyloid and SARS-CoV proteins. So it looks like the virus take advantage of this. You can see here is a blood vessel. So that is accumulating beta amyloid. So it looks like that the virus is taking advantage of this impaired circulation, like by accumulation of beta amyloids to spread. We think that spread faster, or even because we're seeing more presence of that viral proteins exactly when we see this beta amyloid. So I think you're almost time. So I just want to say thank you for everybody that is listening. We have a lot of collaborators to do these projects. I'm not going to be able to say the name of everyone. And I would like to thank everyone to just watching, listening, and take some questions. Thank you.
Danielle, that was wonderful. Thank you so much. Can I just say your images, I mean, the histology it's really beautiful, the images that you've shown us today. So I think it's almost like art, what we've been looking at. Really lovely. We already have a few questions in the chat, and I was hoping to ask this one, but we also have one from one of the listeners. As there are sex-dependent differences that are observed in Alzheimer's disease, we were wondering if there were any also sex differences in your tau pathology or in the astrocyte microglia crosstalk in your model, if you had an opportunity to look at male versus female mice, the primates. Yeah, the monkeys. Yeah.
No, that's a great question. Not yet, but we're doing now. So that's curiosity. I forgot to mention, so that I'm glad that someone asked that. These are all in female monkeys so far, because we have like this old history in our lab to look at the menopause, in fact, and how menopause can affect Alzheimer. So we are looking first at females, and we're just treating females first because we really think that the females might be worse. So we're starting now to inject males as well. So we're going to be able to compare to see if there is a sex difference. I expect there will be something. So, but we still don't know yet. But that's a great question.
And then when you see these changes in the amyloid deposition with age, do you also see any changes in tau, or is it restricted to the amyloids only?
So that's very interesting as well. It's restricted to amyloid.
Okay.
Similar to what happens normally with humans, humans don't accumulate, like we don't have a lot of tau pathology with aging. It's more like to tauopathies or Alzheimer. And it's the same thing with monkeys, like the rhesus macaques. They don't have tau pathology normally with aging. They only have amyloid plaques. And that don't necessarily correlate with memory loss. Sorry.
Oh, okay. Yes, I was going to ask. So if you have been able to characterize the memory changes or the cognitive dysfunction in the monkeys and we're relating it to the amyloid changes, but it doesn't correlate?
doesn't correlate. Similar to what happens in humans, just the presence of amyloid plaques do not cause memory decline in the monkeys.
Amazing. Yeah, that's so interesting. I have a question here as well. Have you characterized the systemic metabolism on these animals while they are aging?
Not yet, but it's something we're trying to do.
Okay.
It's very complex. Yeah.
Oh, I can imagine. And especially the animals are so precious, right? So you have to really prioritize which experiments you're carrying out. What age do you start to see the pathology in the aged macaques?
The amyloid pathology is usually when they're like, I think, between 19 and 20 years old.
Okay.
So it's about 60 something correlating to human age.
So interesting. How similar it is, right?
Very
I mean,
similar,
it really
yeah,
seems
exactly.
very similar.
Mm-hmm.
I'm just trying to see what behavioral tests do you do in these animals?
So far, all this data was done like because we wanted to see some biochemical alteration before doing behavior, because it's just a lot of time, just like 9 to 10 months just to train these monkeys to do behavioral tasks. So right now we are finally looking at the animals injected with the tau mutant, and we are doing some most like memory, like very similar to what we do in rodents. So just like recognition of new objects and just like tracking their activity in the cage, see if they get more mobility with after the injection, but very much like mostly memory tasks to see hippocampus and prefrontal function mostly.
Okay. And we have another question a little bit like that. With your COVID models, were you able to perform motor behavioral tasks after surviving COVID? So did any of their motor function change?
So we didn't do for this first cohort, but we are doing now because 7 days is really fast. We really wanted to see long COVID. So what we've done now is just keep these animals after infection, keep them for longer. So we also be able to look at the behavior. So I don't know yet if there is any change, but it's something we're doing now.
That's amazing. And then as a microglial question, you mentioned your microglia are dying with your AAV tau. I was wondering, have you have an opportunity to characterize what form of microglial death they're undergoing? Have you, or is it an interest in your team to look at the innate immune pathways?
I am we haven't looked at yet, because this is like very recent, but I'm definitely interested in seeing what's happening with the microglia. I don't know, I have this hypothesis that it's getting overwhelmed with the amount of tau and amount of neuron dying, but I don't know exactly what's happening and how it's triggering astrocytes to come and help more with but that's definitely something I want to look now.
Interesting. Thank you so much. We've reached the end of time for this, but I would encourage you, perhaps you can have a look at, I wasn't able to get through all of the questions in the chat, and there's some really lovely questions there. And so we'll now move on to our next speaker. And we have Paige Mumford from the University College London in the UK. So Paige completed her BA in Cognitive Neuroscience from the University of California, Berkeley in 2016, during which she worked in several labs studying different functionalities and diseases of the brain. Since coming to UCL in 2017 for her MSc in Neuroscience, she has been in the lab of Dr. Francis Wiseman in the UK Dementia Research Institute, where they use preclinical models to study the development of Alzheimer's disease in individuals with Down syndrome. And Paige is going to speak to us today, and her talk is entitled Hypersensitive Interferon Response in a Preclinical Model of Alzheimer's Disease Down Syndrome. Thank you so much, Paige.
Great. Thank you, Roisin. Hi, everyone. Thanks so much for attending today's seminar. It's been a really great session, and I've really enjoyed hearing all the talks. So as Roisin mentioned, my name's Paige, and I'm a PhD student and research assistant at the UK Dementia Research Institute at University College London. And I'll be speaking to you about our work on investigating a hypersensitive interferon response in a preclinical model of Alzheimer's disease Down syndrome. So what is Down syndrome? Down syndrome is caused by having an extra copy or third copy of chromosome 21, and it's also known as trisomy 21. And Down syndrome is the most common genetic cause of intellectual disability. Now, there's over 200 protein coding genes on chromosome 21, and having an extra copy of this chromosome leads to widespread disruption in the transcriptome and proteome, which affects the development and function of most organs and organ systems in the body, as you can see by this figure here on the right. And two key features of Down syndrome, which we're interested in our lab, is this prevalence of Alzheimer's disease and widespread immune dysregulation. So what is Alzheimer's disease? Others have mentioned this already, but I'll briefly go through it. So Alzheimer's disease is a neurodegenerative disease that leads to cognitive impairment, and it's characterized by this time-dependent accumulation of neuropathological hallmarks. Firstly, these extracellular plaques made of amyloid beta peptide, and then neurofibrillary tangles of hyperphosphorylated tau protein. Now, there's a significantly elevated prevalence of early onset Alzheimer's disease in individuals with Down syndrome compared to the general population. So all people with Down syndrome will develop amyloid beta plaques by the age of 40, which, as I mentioned before, is one of the pathological hallmarks of Alzheimer's disease. And AD dementia is now the leading cause of death in individuals with Down syndrome. Now, this prevalence of AD in people with DS is thought to be due to the gene for amyloid precursor protein, or APP, being encoded on chromosome 21. So people with Down syndrome have an extra copy of this gene. And amyloid precursor protein, as you can possibly tell by the name, can be processed into amyloid beta, which then can aggregate into the plaques of amyloid beta, one of the hallmarks of AD. So 3 copies of APP has been shown to be causal in ADDS, but we're interested in the effect of the other chromosome 21 genes. So how does having 3 copies of these other genes modify the development of Alzheimer's disease in people with Down syndrome? Now, as I mentioned before, another aspect of Down syndrome that we're interested in is immune dysregulation. And one key component of the immune dysregulation found in Down syndrome is a hypersensitive interferon response. So interferons are part of the antiviral response, and 4 out of 6 receptor subcomponents for interferons are encoded on chromosome 21. So these genes are IFNAR1, IFNAR2, IL-10RB, and IFNGR2. And in the peripheral immune system of people with Down syndrome, there's evidence of elevated expression of these interferon receptor genes as expected, but then also evidence of a activated interferon response, overactivated interferon response at multiple levels of the interferon signaling pathway. So essentially, the immune system of someone with Down syndrome has an activated interferon response, even if they're not actively fighting viral infection. And this has been also seen in a Down syndrome mouse model. So this mouse model has an extra copy of these interferon receptor genes encoded on chromosome 21, as well as many other genes from chromosome 21, and also shows a hypersensitive interferon response in the periphery in response to stimulation. Also, I'll mention that this interferon hypersensitivity in Down syndrome is clinically relevant in that there's an ongoing clinical trial to treat inflammatory skin conditions, which are common in people with Down syndrome, using a drug that blocks interferon signaling. So this interferon hypersensitivity is quite important in people with Down syndrome, but this has mostly been shown in the peripheral immune system. So I was interested in if this interferon hypersensitivity could be affecting inflammation in the brain in Down syndrome. Just gonna take a quick sip of water. Okay, so there is some evidence already of altered neuroinflammation in Down syndrome. So microglia are different in people with Down syndrome. There's an altered microglial activation morphology in the brains of people with Down syndrome, as well as in Down syndrome mouse models that kind of indicate an abnormally activated microglial state throughout life, as well as elevated pro-inflammatory cytokines, again, present throughout the lifespan, even preceding AD. And then when AD develops, the cytokine profile in ADDS is different than that of sporadic AD, so AD in people that don't have Down syndrome. So it's unclear which chromosome 21 genes could be contributing to this altered neuroinflammation, but I was interested in the interferon receptor genes. How does 3 copies of these genes contribute to altered neuroinflammation? Now, aside from Down syndrome, in Alzheimer's disease, the interferon signaling pathway has been implicated in response to pathology. So in response to amyloid pathology, the elevated expression of interferon-stimulated genes, or ISGs, has been observed in microglia surrounding amyloid beta plaques in human AD postmortem brain tissue. And A-beta 42 assemblies themselves can stimulate a type 1 interferon response in vitro, in neuronal and glial cell cultures. And in multiple amyloid mouse models, again, ISG expressing microglia, similarly to has seen in the brain, accumulate around A beta plaques, and then also are coinciding with A beta pathological burden. So interferon signaling is definitely upregulated, although it's unclear whether this is harmful or protective. There's some studies which support both, or either. So overall, though, it has been demonstrated that amyloid pathology induces an interferon response in the brain. And I will mention, while I didn't focus on this in my project, but of course this is also important, interferon signaling has also been implicated in response to tau pathology. So something to keep in mind, although I didn't focus on that pathology for my project. So kind of taking everything I've said into account, I was interested in if the interferon response to amyloid was altered by having an extra copy of these chromosome 21 interferon receptor genes. And the way I asked that is with mouse models. So I used the DP2-TIB mouse model, which has an extra copy of those interferon receptor genes. So briefly, human chromosome 21 has orthologous regions in the mouse genome. And we have a bunch of different mouse models which have been created by collaborators and other groups as well that have an extra copy of groups of chromosome 21 orthologous genes in the mouse. So the idea here is you can map a phenotype that's seen in people with Down syndrome to specific groups of chromosome 21 genes that could be causing those phenotypes. And in the case of my project, I used the DP2-TIB model here, which has 3 copies of a small group of chromosome 21 orthologous genes, so just 32 genes, but within those genes that are in 3 copies in the model is the interferon receptor genes. So major takeaway here is this model has an extra copy of the interferon receptor genes, so I can see how that affects inflammation in the brain. I then crossed this model with the APP-NLGF model. So this is a model of amyloid pathology. It's a knock-in model, so it has a humanized amyloid beta sequence within the mouse APP gene, so it creates human A beta. And then it also has 3 AD causal mutations knocked into the mouse gene. So this model develops a time-dependent amyloid accumulation. So with this cross then generated these 4 genotypes. And of course, the main genotype of interest here is the double, which has the DP2 APP-NLGF, which has both 3 copies of that gene region, including the interferon receptor genes, as well as amyloid pathology. And the key comparison here of interest is the double compared to the NLGF. In comparing these 2 groups, we can see if there's a hypersensitive interferon response to the amyloid pathology when you have an extra copy of the interferon receptor genes. So firstly, I characterize amyloid pathology in these models, basically to make sure that if I did see a neuroinflammatory difference later, it was not due to just a differential amount of amyloid. So starting off with the amount of A beta deposition, didn't see any changes in A beta deposition between the NLGF alone and then the DP2 NLGF. I also looked at levels of soluble and aggregated A beta isoforms, so 38, 40, and 42. And I didn't see, again, a change in the amount of the abundance of these isoforms. Lastly, I also looked at different APP processing products. So full-length APP, CTF beta, CTF alpha, and of course, the models that had amyloid pathology had differential APP processing. But if you just compare these 2 groups here, there wasn't any effect of having an extra copy of those genes in the DP2 NLGF on levels of these different APP processing products. So overall, we thought this is evidence that if you have an extra copy of these genes, this doesn't affect amyloid biology itself, but then later, if you look at the neuroinflammatory response, if there is a differential neuroinflammatory response, it's the response itself that's different and not just a response to different amounts of pathology. So this was an important thing to first kind of confirm. So when I then looked at the neuroinflammatory response, focusing on the interferon response first, I saw a hyper-elevated interferon response in the model with 3 copies of the chromosome 21 interferon receptor genes. So I looked at several interferon transcription factors and found that in the model with 3 copies of the genes and amyloid pathology, you have hyperelevation of these transcription factors. And this was more robust when I looked at the level of interferon-stimulated genes, ISGs. So these are just a few that I looked at, and I also saw the same results in the hippocampus. So you can see here, there's an hyper elevated expression of these genes when you have an extra copy of the chromosome 21 interferon receptor genes. So overall, this is some evidence that we have a hyperactivated interferon response, but our downstream neuroinflammatory pathway is also altered in this cross. So I looked at multiple different pro-inflammatory cytokines, and this time I saw a little effect of having an extra copy of this gene. So this is just 4 of the cytokines I looked at, but overall, pretty little evidence of differences between these 2 groups of interest. I also looked at another component of the immune system, so the complement system, which is also another pathway implicated in AD. And in the Down syndrome brain, multiple components of this complement system have been seen to be upregulated. So this was why I also had a quick look at this pathway, not an exhaustive look at the pathway by any means. I just looked at two of the components, C3 and C1QA, at the transcriptome level, and saw essentially no evidence, no significant evidence of a hyper elevation of these components in the model versus the NLGF alone. So overall, we see kind of little evidence of other broader neuroinflammatory pathways being changed as a result of having an extra copy of these genes. Now, I'll also mention this was all work done by Dr. Gareth Banks at MRC Harwell, so I didn't do any of these behavioral tests, but we also were interested in if there is a differential behavioral response to the presence of amyloid biology in the DP2 NLGF, and with a focus on anxiety. And the reason we were looking at this is that others have shown there's behavioral effects of interferon signaling. So for example, interferon alpha immunotherapy is used to treat chronic viral infections in people. And this treatment with interferon alpha has been associated with the development of anxiety and depressive phenotypes. And in the context of Down syndrome, multiple psychiatric symptoms including anxiety and depressive behaviors are more common in individuals with Down syndrome, and that no one's really looked at, or no one knows why this is more prevalent in people with Down syndrome. So we thought maybe it's the interferon signaling pathway that's contributing to this. So Gareth did 2 behavioral tests. The light dark box test, essentially you have a mouse that has in a box that has a light and a dark bit. And if the mouse spends more time in the light, they're less anxious, because usually a mouse wants to avoid the light bit if it's more anxious. And then the marble bearing test, again, the mouse is just put in a bed with a bunch of marbles that it can bury. And if it buries more marbles, that's seen as a measure more of compulsive behavior. And overall, Gareth did see a few differences in both of the tests. So there's an increase in anxiety behavior in the DP2 NLGF overall compared to wild type. And then in the marble bearing test, there also seems to be kind of incremental deficit in or change in compulsive behavior. But we think we want to do a few more behavioral tests before definitively saying that anxiety is altered one direction or the other in this model. But overall, we feel comfortable in saying that there's a generally perturbed anxiety behavior in this model, which could be potentially due to the hyperactivated interferon response that we're seeing. So to conclude, I used a mouse model of Down syndrome, so the DP2 model, which has an extra copy of the interferon receptor genes of interest. I crossed that with the APP-NLGF model of amyloid pathology. And then in the double cross here, which has both an extra copy of the interferon receptor genes and amyloid pathology, while I didn't see an effect on amyloid biology itself, the interferon response to that pathology was hyperactivated when you have an extra copy of these genes. I didn't see much evidence of altered kind of broader neuroinflammatory pathways, but there is some evidence of perturbed anxiety behavior in the model. And just why we even do this work, why we think it's important, is that differences in the neuroinflammatory response to pathology in people with Alzheimer's disease Down syndrome could impact the use and choice of therapeutics in this population. a population which I'll mention has been historically excluded from AD clinical trials. So we think it's important to understand these differences. Thank you so much for listening, and for Proteintech for giving me the opportunity to speak.
Thank you, Paige. That was really interesting. While we're waiting for some more questions in the chat, I have a few questions myself, because this work it's so relevant. And what I always find striking about Down syndrome research is how similar the pathology is for Alzheimer's disease. And I think then hopefully this will mean then some of the AD therapies that we see that are being approved could then eventually extend across to those with Down syndrome as well. So I was wondering if you'd comment a little bit on your model. Do you see changes in the blood-brain barrier in your double-crossed mice, and perhaps any changes in then the peripheral cells that are accessing the brain?
Yeah, great question. Thanks, Ashin. So unfortunately, everything that I presented is everything that's been looked at in this model.
Okay.
It's a completely new model, but that is definitely something that I'm interested in looking at. As I mentioned before, not this exact Down syndrome model, but a similar model has been shown in the periphery to have hyperactivated interferon signaling in the periphery. So it could be infiltration of peripheral immune cells that's contributing to this phenotype I'm seeing in the brain. I don't actually know.
Okay.
But that would for sure be something interesting to look at. And regardless, I think others, of course, have shown that inflammation in the periphery can contribute to inflammation in the brain. So regardless of infiltration or not, it would still be something really important to look at, characterize the peripheral immune systems and this model. Future work to do.
There's always a long list as well, right? And in terms of the brain itself, do you have an idea or a hint at which cell type might be contributing, microglia more than astrocytes, or have you any ideas there?
Sure, thank you. So again, haven't looked at that, but future work will be, we have a new student that just started actually, who'll be kind of carrying on with this work after I leave the lab. And she's going to look at specific cell types. So I don't know exactly which cell type is responsible. I mean, all of the cell types have the potential to do interferon signaling. They all express the receptors. And interferon responsive microglia and astrocytes and oligodendrocytes have been published in papers, and then also neurons react to interferon signaling. So it's difficult to say. If I were to guess, I'd probably say microglia. There's just more data that microglia are altered in Down syndrome in the brain compared to the other cell types. So that's probably the one I'd focus on first, but we don't know. It would be really interesting to look at.
It would be as well, right? Because I think definitely microglia would probably be my first guess as well. But as we heard earlier on, astrocytes they're so immune responsive in addition. So we have a question from the chat. Have you tried, or perhaps someone in your team, to rescue the phenotype by blocking type 1 interferon signaling? And really great talk, by the way.
Well, thank you. Yeah, that's a great question. So in the future, we'd like to attempt that. So we currently don't have a colony aging, but if we were to restart that would be something we'd attempt. So there are some JAK-STAT inhibitors, so that would block interferon signaling and see if that rescues the phenotype. But that would be something that we definitely want to do. Again, all future work, and it would be, of course, clinically relevant. If that were to be effective in the models, that'd be great.
Absolutely.
Future work.
Yeah. And then perhaps something one could think about is, I mean, you showed so nicely that the amyloid changes are similar between the two models. But I would be curious to see if how the cells are interacting with the amyloid is the same. You know, if they're clustering around the amyloid plaques the same, or if their phagocytosis is similar, because you do see a phenotype, right? So it would be curious to see where that's kind of kicking in at a pathology level.
No, for sure. Again, yes, great point. The characterization of amyloid deposition I did was very, very basic. Is there more or less overall?
Well, you ticked all the boxes, definitely, and the processing is always very important. So yeah, thank you so much for sharing this data with us.
Thank you so much.
We've reached the end of the time for that particular talk. Definitely keep adding your questions to the chat. And I'd like to introduce our final speaker, last but by absolutely no means least. We have Ismail Adeniya. And Ismail earned his bachelor's degree in human physiology from Ladoke Akintola University of Technology in Nigeria, LAUTECH for short, with a focus on reproductive biology and fetal programming. He completed his Master's of Science degree in physiology with a major in neuroscience at the University of Ibadan. During this time, he gained valuable research experience in neurobehavior and neuroinflammation. Currently, as a PhD student at the University of Ibadan, Ismail is integrating his undergraduate and graduate experiences to investigate the effects of maternal immune activation following routine exposure to an endemic parasite on fetal neurodevelopment and neurobehavioral outcomes in offspring. And the title of his talk is OLAX Subscorpiodia, Prevented Scopolamine-Induced Memory Impairment Through the Prevention of Oxido-Inflammatory Damage and Modulation of Cholinergic Transmission. Thank you.
Thank you for introducing me. Can you hear me?
Yes, I can hear you.
Okay. I think by the end of my talk, I will be able to convince you that natural products have a place and a role to play in the case to tackle some major neurodegenerative disease, especially Alzheimer's disease. So as earlier said, I will be presenting on a natural product named OLAX Subscorpiodia, and its role in preventing neuroinflammation damage and modulation of cholinergic transmission in Alzheimer's, in a scopolamine model of Alzheimer's dementia. One thing that we've been able to achieve since the turn of the 20th century is to increase life expectancy. So, and in our case, to maybe later on, if we can get to immortality. So, but with that increase in life expectancy, with increase in medical healthcare and services, is the increased risk of age-related neurodegenerative disease, with Alzheimer's dementia playing a prominent role in this neurodegenerative disease. So currently, it's projected that about 50 million people suffer from Alzheimer's dementia, and the population is expected to triple by 2050. So when you observe the brain parenchyma of people with Alzheimer's disease, the classical observation is the deposition of amyloid plaques and aggregation of tau protein forming intraneuronal fibrillary tangles, so which leads to neuronal loss and the loss of cognitive function in people with Alzheimer's disease. And amyloid plaques have been theorized to play the prominent role in the pathogenesis of Alzheimer's disease, leading to neuronal loss. However, recently, early plaque-independent neuroinflammation have also been indicated to play a role, so which has featured as potential drug targets in the management and in the prevention of neuronal loss and improvement of cognitive function in people with Alzheimer's disease. So currently, Alzheimer's disease have no cure. So, and some of the drugs used in its management comes with their side effects. And one of the most prominent drugs that was started to combat the disease, which is a amyloid beta antibody. So failed clinical trial, which means there is this case to there is the case is there, and interest is there to look at maybe drugs in its management and its prevention. So in our laboratory, our focus is in the prevention of this early plaque-independent neuroinflammation before leading to Alzheimer's disease. So one of the things that we're interested in is in looking at natural products that are used within the environment. There are maybe people that has been reported that is being used with people around us in Nigeria here in management of some cognitive disorders, and also there is this growing use and demand for natural products, especially since WHO reported that about 80% of the people globally depend on natural products as their primary line of care. So also, there have been progress and some prospects in use of some phytoestrogen, as seen by work conducted by Professor Robert Brenton of Arizona University. So they created some phytoestrogen in their lab that have shown prospects in preventing menopause-related cognitive declines in women. So, and this phytoestrogen factors, phytosan, is currently on a clinical trial, which means there is this prospect for the use of natural products in the management of Alzheimer's disease or cough. Okay, so and also when you look at with the growing interest in the use of natural products, you can see that the economy is also there, which means in addition to the health benefit that we get from the use of natural products, which is readily available in some instances, there is also the market is there, the economy is there in order to also improve GDP across the globe. So, and our natural product of interest there is OLAX. So, and it's very common within West African region and some part of sub-Saharan Africa. So, and also some preliminary studies have shown that the natural product contains some phytochemicals, both the ECRs and the ethanol extractions, and also some methanol extraction of the product have also shown to contain some relevant phytochemicals that have been shown to improve cognition. So, and also the plant is also used in Southwest Nigeria, where our laboratory is by people in the management of cognitive-related disorders. However, there is no scientific basis for its usage, and its toxicity is also not known. So, also some previous studies have also shown that the natural product have potential in modulating apoptosis, and also it has also been shown to possess some anti-nociceptive and anti-inflammatory effects. And also an in vitro study has shown that the extract from the plants has potential in modulating acetylcholinesterase and butyrylcholinesterase, which are enzymes which are also of interest in some drugs like donepezil in the management of Alzheimer's disease. However, this is an in vitro study. So, with that background, what we were interested in our lab is to look at this extract in vivo in plaque-independent neuroinflammation as a prospect in management of degenerative disease. So we use a model with scopolamine model, which has been shown to also drive this early plaque-independent neuroinflammation. So in rodents, and also so we use a methanol extract for in this study. So since methanol extract have been shown to produce high yield of phytochemicals. So after treating the animals with extract and the scopolamine, so we subjected the animals to some cognitive tests. So after the treatment duration of 14 days, so we used elevated plus maze and novel object recognition test. I think Jacob explained how the novel object recognition test works earlier, so I will be dwelling on that. So, but mostly people use elevated plus maze to assay anxiety. But in here, we use the spontaneous behavior of the rodents to seek secluded environment. So we train the animals to seek the secluded environment. Then we look at how far they've learned, they've acquired the memory to locate the closed arm of the elevated plasmids at the trial phase. So, and what we observe is that our scopolamine model spent a longer time to locate the closed arms of the elevated plasmids, and our treatment group of OLAX showed a reduction in the time it took them to locate these the closed arms. So, and especially at the 50 milligram per kg. Also, on the novel object recognition test, the scopolamine model showed a negative discrimination index, which shows that it's poorly differentiate between novel objects introduced at the trial phase, while the treatment group showed improved cognitive discrimination index by as shown in especially the 100 milligram per kg, and including our treatment group, which is donepezil. So, in addition to that, so when you see neuroinflammation usually drives positive stress. So with that, we look at the redox status in the brain or in the samples of the rodent study. So using MD as marker of lipid peroxidation and also nitrite as marker of nitrosative stress. So our treatment group also helps in suppressing lipid peroxidation. When you look at the nitrosative stress, the treatment also suppress the nitrosative stress also. We also look at the antioxidant markers to balance in confirming the redox status in the brain. So what we can see, which is also in consonant with some other studies using some natural products also, is our OLAC also improve GST, which is an endogenous anti-peroxide dismutase. It improves the activity of the level of peroxide dismutase. So in further to that, like I mentioned earlier, what we're looking at is the neuroinflammation. So with that, we look at TNF-alpha, pro-inflammatory cytokines, and also interleukin-6. So what we can see, what we observe is that our treatment group also perform well better when we compare it to the scopolamine group, in the sense that it was able to suppress pro-inflammatory cytokines, TNF-alpha, and also interleukin-6. So we also look at acetylcholinesterase activity. Like I mentioned earlier, some of the drugs used in this management, like the Nepezi, drugs that target the acetylcholinesterase, they inhibit the activity of the acetylcholinesterase to increase the level of acetylcholine at the synaptic cleft. So what we observe is that our product here also suppress the activity of acetylcholinesterase here, showing a prospect in also, which show that it has prospect in also modulating the activity of acetylcholinesterase. So we also do some histology study using H&E, that's hematoxylin and eosin, and also we also did cruciferous violet. So, and in our H&E study, what we observe is that our treatment group actually prevented pythinosis and karyolysis in the hippocampus, especially in the CA3 region of the hippocampus, which is very important in memory acquisition and retrieval. So as you can see by the right also, it also protects neurons against the inflammatory insults that are produced by the scopolamine, which means it's increased the number of viable neurons that is present in the hippocampus of the rodents in this study. And also the same can be seen also in the cruciferous violet, which is the nasal staining. So we also observe that our product also prevented central chromatolysis in the hippocampus. So as we can see here, which is also, there is also increase in number of viable neurons seen in the hippocampus. So in summary, all we see is that our product also has the potential in improving both spatial and non-spatial memory in this study. And also it was able to modulate neuroinflammation triggered by the administration of scopolamine, and also modulate acetylcholinesterase activity, and also promote neurogenesis. So with this information, we can simply tell people that are currently use this product in the management of neurodegenerative disease back here in Nigeria, that's okay. From our study, we know this is safe, or this is the safety index where you can use this particular product and you get this particular response. However, there is also need for further study to look at maybe in isolating some potential compounds that are responsible for the results seen, or to also compare if it is the synergistic influence of the phytonutrient present in the extract that performs the observation seen in this study. So I'd like to acknowledge Proteintech for the platform for the exposure. Actually, this is my first time, and also my PI professor, and also Professor Mahmoud Bukamaina, who has been very helpful towards this presentation. Thank you.
Thank you, Ismail. That was very interesting. I really enjoyed your talk. I have a couple of questions myself actually about this. I thought the effects were striking with the OLACs, and I really liked that you had your donepezil control group as well, so that we could see the comparison and contrast between the two different treatments. Did you have an opportunity to look at microglia in your model to see, because IL-6 is, well, IL-6 would be more of an astrocyte, I think, cytokine, but TNF-alpha are strongly released by microglia. And I was wondering if you saw a lot of gliosis, perhaps, or any changes with your OLACs treatment.
Actually, we are interested in that. This is just a preliminary work to know that, okay, we can get this response. So that's suggested to further study to look at the microglia cell. So actually in our lab, we do IBA1 staining to look at the microglia effect, and also GFAP for astrocytosis. So, but currently that is suggested to actually feature work in.
Yeah, absolutely. And maybe GFAP as well for astrocytes if you have some tissue left over. I think it would be so striking to see the changes. And I missed it actually a little bit with your setup of your experiment. So when you induce this scopolamine, when do you start the treatment afterwards, or is it kind of at the same time?
No, actually, since it's a preventive study, what we do first is to give the OLACs. So so for 7 days, then on the heat, we started co-treatment with the OLACs until the 14th day.
Okay. Well, that's amazing how protective it was. Absolutely. And do you have an opportunity to see, do you know if it's working more in the periphery or if it's accessing the brain, or how would you test that?
So actually, what we do is just to look at how systemic inflammation drives inflammation within the brain. So that's why our focus is just the central inflammation, and that's why the brain is the organ of choice in this study. So currently we are not really looking at the peripheral inflammation. However, there are some work that has been done also with the same product, which is OLACs, to look at the peripheral inflammation, especially a TRICE study, although it's still an unpublished work. So and it's and that's why it's actually very positive.
Yes, I could imagine perhaps if you were able to maybe take the serum or the blood from your animals when you're taking them down as well, it would be, you could compare and contrast perhaps to see where you're seeing your changes. But thank you so much for sharing this preliminary data with us. It's really promising.
Thank you.
Yeah, and I'd like to thank all of the speakers as well for their amazing talks. I can just say for myself, I've learned so much from this session. I thought it was wonderful, all the different angles and aspects we've heard from microglia and astrocytes, and just the different models as well, Alzheimer's disease and Down syndrome and alpha-synuclein related pathologies as well. So thank you very much to Rebecca and Proteintech for organizing this wonderful seminar.
No worries at all. And thank you so much, Roisin, for hosting. You've done an absolutely fantastic job. I'm just going to share everyone's contact details who spoke today. Hopefully that comes up now. And yeah, thank you so much, everyone, for talking. I learned a lot still. Really fascinating talks from everyone. I did my PhD in circadian, so it's really great to hear from Erik everything that he's been doing, and really fascinating to hear. And also just Danielle, those images are absolutely incredible. But yeah, amazing talks from everyone. You can find their information on the screen here. So if you would like to contact any of them, please do reach out. They've all got various social media. I know people in the past have actually, formed collaborations from these events and had amazing collaborations from just this incredibly global experience, which I always think is like really fascinating about Zoom and webinars and everything, that you can sort of hear talks from all over the world without having to really leave your lab, which really helps. But yeah, thank you so much, everyone, for joining. Myself and Proteintech will be at SFN next week if anyone's there. Do pop over and say hello if you're attending. It'd be great to see anyone that was there. And yeah, massive thank you to everyone again. We will send a recording out and also a certificate of attendance, as well as the opportunity to have a free sample. We'll send a follow-up email a few days after this. But with that, I'd like to say thank you so much to all the speakers, and yeah, enjoy the rest of your day, whatever time of day it is for you. And yeah, speak soon. Bye.
Sorry, Rebecca, that's not my email.